So this is all man-made. This is an engineered feat in how to control expression. So what we can do is also do something very similar, is not only to put the promoter up front and redesign this, or take advantage of the IPTG, right, and put back and use lactose as well. So whatever we want, we have to link these two up. So in the bacterial cell system, it mimics the repressor proteins in which it's sensitive to IPTG. But we also can modify this plasmid, having exactly the same thing as the lactose, or use the system that already exists. Okay, so let me repeat this. The system, this plasmid system, has the cloning sites. We have the ribosomal binding sites, and these are all handouts given to you. All right, so these are important. T7 promoter sites, the coding sequences, terminators, and points of origin is what we talked about. It's important. But the sequences for this, up front, we said that this promoter here that's up front to the open read frame. The question is, can we replace this with the entire E. coli regulatory lactopron promoter and have the same effect? Right? And so the trick is, now you have this whole gene. You have the ribosomal binding sites. If you have the regulatory regions for this, and downstream to clone in the green fluorescent protein, can you actually take this whole plasmid, put it in, and have the same regulations, the same regulatory regulation? All right, so what that means is that what you're going to have is a repressor protein that's being made. You have to have an operator and inhibitory sites, a promoter, and a downstream green fluorescent protein. Basically, pop that whole thing that we've talked about before. And now you put it into an E. coli system, and as long as this plasmid is stable, meaning that it doesn't leave the E. coli, it should be able to have the expressions of these. Right? Okay, now, your job is to start now expanding your thoughts in terms of, you made the green fluorescent protein, the sequences, right? And so you have an idea of the opening frame. You look for restriction sites inside that may impair the entry of these. And now what you want to do is now look at the lactopron promoter region and the operator region. Look up the sequences for those. Right? So those sequences are available now in the NCBI in a lot of these banks. And so I'm going to leave that up to you. We're going to pick up this discussion on Wednesday. Right? So what I want you to start doing is to look up the promoter regions, the upper regions that we talked about in the lactopron, the repressor protein sequences, and find out what these sequences are. Right? Now, in terms of organization, I want you to start doing this. Get a folder. All right, we're going to start compiling bio parts now. The first bio part right now is purely digital. All right, take your open green frame, your green fluorescent protein, and print it out on one page. You can color it green or whatever you want to highlight it. Okay. All right, and that only label this is open green frame, green fluorescent protein. You could have a chapter title on that. Okay, now next one are regulatory regions, promoters. Right. So your first one is to find the sequence of the lac promoter and operator site. Right. And for that matter, find the sequences for the inhibitory and get a sequence of that and outline clearly what are the sequences that are necessary. So here precision matters. It's no longer just drawing boxes. We're talking about every letter now, where it cuts off, where it begins. All right? Because if you miss a letter or it's a wrong letter, that will destroy the whole system. So you've got to be very, very careful. right? So what I want you to start doing now is expanding on what we started out with. You got an open ring frame, that's your first valve part. Now second one is to work on this promoter, the operator, because this is one of the regulatory system. And what we're going to do is design that and make that as a separate system. All right. Now later, what we're trying to accomplish is this: is get a list of a regulatory regions of our interest and open reading frames of our interest. Right. But and what I have you assign is to not only look for green fluorescent protein, but for other proteins that may be of your personal interest. So let's say the antigen for breast cancer, BRCA type proteins, right? Even though what we're going to do in this class is the green fluorescent protein, but at least to build up this library is to have open ring frames of your individual interests. At the end of this course, I'm going to have probably work in groups of 2 and have a particular unique library of bioparts of their open ring frame and controlling sites, operator sites. But what we have in common is we're going to use the green fluorescent protein, mainly because it's easier, it can be done in a short period. But what I want you to start looking at is think about the operating sites that you want. And what we're starting out as a group is to work on this LAC operon. But the promoter sites is what I want to differentiate among all of you. And this probably may divide in 2 or 3s. Are you interested in a promoter that maybe are sensitive to heavy metals, for example, or toxicity? Because what we're trying to do is use green fluorescent protein as a marker, and the promoter site, the upstream regulatory region, are what is sensitive to whatever's environment. So the question is that in the presence of X, green comes on. That's what we're trying to do, right? So just to get you into the system and get you rolling, we're going to work on the LAC operon. And hopefully we can construct that and show you that in the presence of lactose, we could make this thing turn green. Okay? Any questions up to this point? All right. You see where this is going? We're making bits and pieces by parts now. And so now we're assembling genes, which eventually will lead to a pathway. All right. Now, synthetic biology, as I defined it, could mean 2 things. It could mean artificially modifying the organisms in which we are doing, or, okay, which what we're not doing, is creating a completely new organism. That's a whole new animal. So usually when synthetic biology is discussed, it's usually discussed in these 2 frames. For this course, it's modification of pathways and so forth. We're going to leave the creation of the organisms to Craig Venter, right? We're not going to do that. But at least this gives you a start of practicing with bioparts and things like that. Okay, any questions before we leave?
I'm still a little confused about the IPTG.
Okay.
You say it looks like the other.
Yes, so if you take the LAC off
Do we just turn this off?
yes, please, just turn it completely on. So if you take the LAC operon, and in the presence of lactose, this gene will be expressed on. A common thing is that you want to do exactly the same thing, except use IPTG. And the reason is several-fold. One of them is that IPTG does not get cleaved. It doesn't have the glucose component, right? But the repressor recognizes more of the galactose moiety. And what it does is that it will block the repressor, it will turn on the gene, but it will not degrade. That means it will stick onto the repressor.
Okay.
In the lactose, once it breaks, the enzyme comes in and it's gone, and then it turns off the gene again. So it's a type of regulatory system. If you want something that's constantly on, this is much more stable, so it's a permanent bind. And so that's one way to keep it continually rolling. The second thing is that in terms of what we're trying to do, you want to control it very well. A lot of the a lot of recombinant expression system has what is called leakage. And leakage implies that you have proteins that are expressing at very low levels. Even though you don't have an induction going on, you still have low levels of expression. And you don't want it to express until you tell it to. And so this is a very clean way to drastically turn things on and off. And this may not be pertinent for green fluorescent protein, but if you're expressing a protein that's toxic, that the E. coli doesn't like, but you want to produce it, you don't want residual expression. You'll kill your bacteria before you get to a prevalent production level.
Okay.
And as far as the stability thing that we were talking about at the beginning with the, sometimes the cells will just eject plasma.
Yes.
Is it based on like metabolic resources, like the cell just sees, okay, this is something I don't need and I'm just gonna get rid of it?
Yeah.
Unless it's like exposed to ampicillin and it's like, okay, well you're gonna keep me alive, so I'll keep you around.
Well, small chromosomes are unstable. They either degrade or they don't hang around unless there's some kind of constraints to them. For our purposes, it's kind of the opposite of what genetic engineers want. We kind of don't want the stability because in case we want to make something, we only want to make it that lasts us maybe 1 or 2 generations. Okay, and after we want that plasmid to lose it because we don't want this to be hanging around. We don't want a permanent strain on this. And so again, green fluorescent protein is pretty much harmless. But if you're talking about having something a little more toxic to the environment, or you think that has some unknown entities that may be dangerous, you don't want it. You want to have a way to have it self-destroyed or not making it. So even if you can't control it, for example, it'll at least
It'll naturally just
peter out.
Okay,
for lack of a better word.
And these, is it just like by the nature of the plasmid vectors, like these engineered vectors, that they're going to do that anyway? It's
Yeah,
just kind of by nature?
yeah, and like I said, this is a problem you don't want to have
Yeah.
in the lab because a lot of the problem you make a recombinant expression protein, you want it to last forever, and you want to keep this in your freezer. The problem we have is that if you, after a few generations, sometimes the plasmid is gone and you lose that, and that's why it's for genetic engineering, you always want the plasmid itself as a backup system, right? Not in the bacteria, because the bacteria will lose it. But in our case, it's a good thing because we don't want to make it permanent, and that's how we're defining it. We want to make something that is absolutely not permanent, but at least we have the construct. So you have the bio parts set aside. We have now an open reading frame. I want you to consider other open reading frames. The regulatory regions, we're talking about the lac operons now, and the ones we're going to construct are the lac operons for green fluorescent proteins. But later on, I want you to accumulate a library of bioparts. And we're only talking about open ring frames right now, and we're only talking about promoters and regulatory regions, all right? And we're going to go through all the necessity. We're going to talk about the ribosomal binding site and the terminator. Right, so those are the ones that's left that's going through this. Okay, I'll see you on Wednesday. Please start looking at the lac operon sequences. Find out.